Good afternoon. My name is Kane. I'm senior. It's my pleasure to introduce our next guest from ReCode, President and Chief Scientific Officer, Dr. David Lockhart. As a reminder to attendees, if you'd like to ask a question of our speaker, please type it into the question box under the video player. So David, welcome. Thank you so much for joining us today. And maybe to start, can you provide a couple of minutes of introductory comments about ReCode for investors new to the story?
Yeah, thanks, Kay. Happy to. So at ReCode, we're a precision genetic medicines company. I think importantly, we have two homegrown messenger RNA LNP therapeutics that are in the clinic already. And both of those were developed using our own next-gen LNP delivery platform. And we'll probably talk about it more as we go along, but this is a very broad and versatile platform that allows us to package a very wide range of genetic medicine payloads, mRNAs, base editors, prime editors, and so on, multi-component cargoes all together in the same LNP. And in addition to being able to package that wide range of payloads, we've shown that we can deliver beyond the liver and that we can do that with multiple different routes of administration. So we think a very enabling platform for being able to do a very wide range, for developing a very wide range of genetic medicine drugs.
Well, great. Well, David, I think most of our audience appreciates that LNP for nucleic acid delivery typically consists of four lipid components: an ionizable aminolipid, a structural phospholipid, cholesterol, and a PEG lipid conjugate. And because of the affinity for ApoE that gets stuck onto it during its journey for the LDL on the liver, that's where most of the classic four-component, let's call it MC3 type LNP goes. So what are you doing to achieve the tunable performance with your sort design?
Yeah. So we also use the core four components. We have proprietary versions of the ionizable lipid, but so the biggest difference is that we add a fifth or we add a sixth. I used to be able to do it with just one hand, but now we're using even 6 component LNPs. And it's the addition of these chemically distinct lipids beyond just the four that allows us to tune a lot of the very important properties of the LNPs. And so with the right fifth or sixth lipid in the right relative amount, we're able to do things like deliver systemically and avoid the liver, as you said, instead of going to the liver because they bind ApoE. Instead, we can make them not bind ApoE so that the liver is not a sink and they can deliver payloads elsewhere in the body. And also now we've found with other routes of administration that, again, the same type of fifth and or sixth lipids allow us to tune the cell tropism properties. So even when we're delivering more directly, as we are for our lead programs with an aerosol delivery directly into the epithelial cells of the airway, even there we use a fifth and or a sixth lipid to tune the cell tropism so that we deliver to the cells that are most important for the diseases we're going after. And as you know, even for genetic respiratory diseases that are related, where the target cells are epithelial cells that line the conducting airways, for diseases like cystic fibrosis and primary ciliary dyskinesia, it's not the same subset of cells. So that ability we avoid the liver just by delivering as an aerosol, but then we can bias the delivery strongly towards the cell types that are critically important, and we don't waste drug or deliver extra lipid to the cell types that are not important for the effect that we're trying to achieve. So I think that those are really the key differences: is the addition of chemically distinct components that really give us levers for tuning the properties that you don't normally have with just the core four.
Great. And how different then, if at all, are the LNP formulations that you're utilizing for your first two lung-targeted candidates, PCD and CF?
Yeah. So they're related. I usually refer to them as cousins. They actually share four components. They share the ionizable lipid, but they differ in those the sort lipids, those fifth and or sixth components, because those are the components that we've used to differentially tune the cell tropism profiles. So they have a lot in common. They're similar in size. They have similar surface charge. They both can efficiently encapsulate the messenger RNA. They both can be nebulized well using mesh nebulizers. So they have a lot in common, but they differ in those key components that give us the tunability.
And why did these first two lung targeted candidates represent the best place to prove out the ability of your sort to really effectively deliver to targets outside the liver?
Yeah. It's a great question. There are, of course, lots of other targets and diseases and indications that are of interest to us and that would have been interesting to work on. Really, for the two that we pursued first, there were several factors. One is, in both cases, we're addressing populations that currently have no therapies or no disease-modifying therapies, so clear unmet medical need. Population sizes that are big enough to run clinical trials and for eventual, commercial viability. And also, this allowed us to, what we liked about these first two is that the target cells are known. So the target is known, so there's no target risk, but also the target cells are well defined. So with an aerosol delivery, we know that we were very confident that we could deliver to the cells that mattered in the disease, while at the same time minimizing the systemic exposure. So one reason to go after diseases of this type with this mode of delivery first is that this is the first time these types of LNPs have ever been in human beings. And it made sense to us to deliver them as specifically as possible to the cells that are important and minimize overall systemic exposure. Of course, there's nothing limiting about our LNP platform. So we're not limited only to delivery to the lungs. We're not limited to only aerosol administration. I think, as I mentioned when I was talking about the company, we've now shown that we can deliver IV, which is where the technology was originally developed, deliver as an aerosol to be inhaled. We've delivered intramuscularly in the context of mRNA vaccines. And we've shown that can elicit a strong immune response in rodents. And then more recently, we have shown that we can deliver intrathecally for delivery of genetic medicine payloads into the parenchyma of the brain. So those are the ones we've explored so far. So we can continue along that path of other routes of administration. But I think it's important that we thought we made a good choice given what was known about the diseases, the clear unmet medical need, the targeted direct delivery to the most important cells, but also understanding that the technology has a lot of room to grow beyond that, taking into consideration different modalities, different routes of administration, and the differential ability to tune the biodistribution properties and cell tropism profiles.
Okay, and maybe as a bridge to taking the discussion to CMC, there was a previous effort by another company, Translate Bio, to deliver an mRNA therapeutic to the lungs to address CF. And what were you able to learn from their experience?
Yeah, we actually learned quite a bit. We studied their program quite carefully, and they published a very nice paper on the program that they took into the clinic. Really, the key K is that they didn't have that tunability. So they had a more traditional type of LNP. It was actually a 3-component LNP where 2 of the components are covalently linked, but overall chemically very similar to 4-component LNPs. And they didn't have the ability to tune the cell tropism. And they, during the preclinical development, they didn't show that they could deliver specifically to the cells that mattered most, and to do that all the way up to non-human primates. And then importantly, they also didn't show that they could restore CFTR function in the gold standard human cell model. They did show some restoration of function in the FRT, the rat cell model, but not in the human cell model. So we learned from their experience to make sure that we showed that we could deliver to the right cells all the way up to non-human primates to get a strong response in the gold standard human bronchial epithelial cell assay. And then also we learned that it was very important to make sure the LNPs nebulized well. And so for the Translate Bio program, at the doses they were using, they had nebulization times that were as long as 2 hours. And our nebulization times at our target doses are only 5 to 10 minutes. So I think that was another key factor, is very early on, in addition to all the other properties, we made sure that the LNPs nebulized well using standard commercially available mesh nebulizers, and that they didn't fall apart, that they didn't clog the nebulizer over whatever period of time we needed to nebulize, and that we kept the nebulization session short. When we were first thinking about these diseases, we talked to the KOLs, treating physicians, the patient groups, patients, and all of them said nebulization is fine. People with PCD and CF often use nebulizers for the delivery of other agents, but that was fine. Just don't make it a long session. So very early on in our TCP, we wrote in 20 to 30 minutes or less. And with optimization and so on, we were able to reduce that time even more. So I think that was another important lesson to really focus on that early, so that we could do something that would be as convenient as possible for the patients.
Okay, so with all this learning in hand, you proceed to design your candidates. So from a CMC perspective, for these first two inhaled mRNA-based therapeutics, what are some of the key challenges you had to overcome?
Yeah, these are, as you know, LNPs plus mRNA. That's not an easy CMC problem. So the positive part is all of the components are made with either pure chemistry or cell-free biochemistry. So that works in our favor. The other thing that really works in our favor is that the methodology for formulating LNPs with mRNA really advanced because of the COVID experience. And so we were able to take advantage of that. And so even though our SORT LNPs have sometimes 5 or even 6 components, the formulation process is the same, very similar to what's used for the COVID vaccines and for 4-component LNPs. So we really got to take advantage of all of that work. And then, over time, it's also the case that just the scale up of making mRNA and making it at very high quality, and the analytics that go along with the RNA part of the process. Again, those all have advanced over the last years with the big boost from all the work during COVID. And so we've been able to take advantage of all of that work on both the mRNA side and the LNP side and the formulation step, as well as all the methodology around purification and the analytics that go into making sure that you really have a well-defined particles with good encapsulation efficiency, with RNA that has high integrity, and so we just kind of learned all of those lessons, and we paid attention to them very early, because I think in this game, it's not an easy process, but also it's clear that the quality of the materials can make a night and day difference. The purity, the characterization, the consistent size. And we paid attention to that early, and we implemented processes that made it so the way we made materials at small scale was essentially identical to the way we made it as we scaled up. So each time we scaled up, now at this point, over more than 3 orders of magnitude, from the 5 milligram scale to greater than 5 gram scale, the processes we use are very, very similar. And as a result, we've been able to scale quite, it's never straightforward. These materials aren't straightforward, isn't the right word, but it has been pretty much major hurdle free. We've been able to scale because we use processes early on that could be scaled. and we've been able to keep them consistent.
Well, great. So it sounds like, you know, your advantage by your early focus on CMC. Obviously, you've gone through reviews of your INDs, you know, I guess two-part question. One, was there any pushback from the FDA on any aspects of your CMC during this process? But obviously, as you move forward to let's say a BLA review, where there's going to be a much more exhaustive and thorough evaluation of your CMC, you know, how are you feeling about the strength of your analytics, your purity, your potency assays? You know, how are you measuring potency if the end result is to produce a protein? So talk about that.
Yeah. So how am I feeling about it? I'm feeling pretty good. The response from regulators was thorough, which I appreciated. I think they've learned a lot, you know, over the previous years from the COVID vaccines and the Translate Bio experience, and you know, Vertex Moderna experience with their CF mRNA program. So they're clearly putting all of that knowledge to work. No major red flags. I think they're just being extremely careful because these are multi-component agents. And so they're paying a lot of attention to purity and source materials and, you know, all of the documentation that goes with those things. So all appropriate. And you really hit on an important point. Really kind of the area that is a little less defined is on the potency assay. So, of course, we do all the physical characterization, you know, size and surface charge and encapsulation efficiency and purity and all of those things. So everyone knows to do that. But for a potency assay, there's no one size fits all, because the potency depends on what it is you're trying to do with it. And so that can involve, you know, different types of cell-based assays. So we've really focused on cell-based assays for the potency. And those, in some cases, can be measures of actual function of what we're delivering. And in some cases where that is not practical, measures of at least the encoded protein being made at significant levels and consistent levels. So really, you know, so I think that's important. So cell-based assays, well-chosen cell types, methodology, of course, but then also either a functional readout, if that's possible, or something that at the very least shows that the mRNA got delivered, got into the cell, and then got translated into the right protein.
Okay, great. So while ReCode is currently utilizing a CDMO, how is the company thinking about scaling up manufacturing to commercial levels?
Yeah. So I think another thing that has worked in our favor is that, so you're exactly right. So for our material that's in clinical studies, that's made under GMP with CDMOs. So that makes sense. But we ourselves have done full formulation synthesis of the materials up to comparable scales. So when we did tech transfer, we were doing tech transfer of processes that we had done in our own hands at a very similar scale. I think that really helped when we were doing the tech transfer and to make that as smooth as possible. We also now have done internally, we have done production of material that we would call GMP-like, that is suitable for doing tox studies, so GLP tox studies. So we're not making material for going into human beings yet internally, but we are making material that can go into GLP tox studies that will be part of regulatory filings. So really the question about what we do when we are going to the next step is really just a matter of kind of just cost and speed. There are some really good CDMOs in the world for making mRNA and LNPs these days. That's been another benefit of, you know, the flourishing of the field over the last 5 years. So we have the option, but we do have the expertise and we do have the option of doing it ourselves. And it'll kind of come down to just practical matters of cost and speed when we're at the point of having a launch, you know, in our near future. So we do expect to continue to use CDMOs for the foreseeable future. But I think given all the things I said, we're not locked into that if it makes sense to do it ourselves, just because we have been able to produce material at scale under GMP-like conditions already.
Okay, great. So I know you touched on something earlier, so I'd like to explore. And so from a broader perspective, what are some of the important lessons learned regarding the manufacturing of a mRNA-based therapeutic using LNP from the COVID-19 vaccine experience? Obviously some clear caveats, though. Those are vaccines designed to provoke an immune response. You're delivering a therapeutic. You want the immune response to be silent. And those are designed to be given, less frequently, years more frequently. So talk about that.
Yeah, so I like I said, I think we got to learn a lot of great lessons in terms of being able to produce these agents at scale, and regulators got a lot of experience, reviewing programs of this type and figuring out what it was they needed to see. And then you're exactly right, Kay. We've just taken it steps further, because now we're going to be delivering, in some cases, higher doses and definitely much more frequently for the lead programs. When we get to programs for gene editing and correction, then the dose frequency could be one and done, or very infrequent and done. But at least for our mRNA replacement programs, there we expect to deliver pretty frequently. So basically, all of the learnings around purity and analytics, we've just taken to a greater extent to make sure that the agents have very minimal immunoreactivity, as you said, that they have very good purity. And that, of course, that we put them through very rigorous testing in the GLP tox studies, so that we make sure that we have a good therapeutic window, and that repeat administration is well tolerated. So I'd say we just took all of those lessons and just knew that we had to do even more because of the things you said with regard to dose and dose frequency.
Okay. Let's talk about COGS, because clearly, relative to some of our other presenters today, manufacture of mRNA LNP is less complicated. Not that it isn't complicated, but less complicated than relative to cell therapies and gene therapies. That said, lower COGS is always an objective. So how will this be achieved in the manufacture of mRNA LNP therapeutics?
Yeah, we think it's a combination of a few things. I mean, one is just scale. So there are going to be economies of scale. But there are also other places where we think we can drive it down. The mRNA, making the mRNA is a significant fraction of the cost. And so I think improvements there will come on several fronts. One is just optimizing the design of the mRNAs, so that of course they still encode the protein that we want them to encode, but with optimization for in vitro transcription yield, for physical stability, for translation efficiency. So all of those could help us make more material for the same cost. And then also, if they are more physically stable and have a higher translation efficiency, the dose can come down. So that will save on the COGS as well. And so I really think a lot of it will come on the RNA side, really optimizing the mRNA itself, but then also just improvements in the enzymes that are used to make the RNA, just ones that are more processive and that have higher efficiency. And then on the LNP side, it's kind of more of like a standard chemistry sort of problem, where you can just being able to scale chemical processes is just a matter of doing the work. And pharma companies and have been doing that for a long time. And I think there's significant room for improvement there, just being able to make batches at larger size and be able to do it with less expensive processes. So that'll just be part of the process optimization. But I think the biggest savings are likely to come on the RNA side, and being able to lower the doses so that we just don't have to make and deliver as much material either.
Okay, well, let me squeeze in one last question here,
Okay.
David. Can you outline the key milestones for the company over the next 12 to 24 months?
Yeah. Well, this won't surprise you because we're a clinical company now. So a big part of our milestones involve moving those programs forward and hopefully getting promising data. And I think one thing that is important for our programs is that even though we've only been in the clinic for a relatively short period of time, we have readouts coming that are very meaningful this calendar year and next. So much so that in 2024, if things go according to plan, we could get efficacy data in both cystic fibrosis and PCD patients showing that the delivered mRNA is working, that it's restoring the biological function that we're attempting to restore. Those studies this year, at the same time, of course, showing safety and tolerability. So we have studies reading out for both PCD and CF this calendar year. They're not fully powered, so we refer to those as proof of activity clinical studies, which then set us up in 2025 to do proof of concept studies. And when we say proof of concept, we mean studies that are fully powered with approvable endpoints, at least with endpoints that could be the basis of accelerated approval.
Right.
So that's pretty exciting. So even though we're relatively new to the clinic, the path is pretty fast if things go well, where we can get clinical efficacy data this year that sets us up for studies next year that could actually be the basis of approval, because we're using endpoints for cystic fibrosis that have already been used, such as FEV1, and we're doing very analogous things. Even though there are no drugs for PCD, we're doing very analogous things for PCD, where we can use FEV1 and similar supportive endpoints as those that were used for CF. So I think, and these are rare disease trials, and we have a good basis for doing the powering calculations. So we know that we don't need a very, very large number of patients. These are like the trials that Genzyme and others run, with 40, 50 or so patients. So all of that really works in our favor, that we have a fast path into the studies that could be the basis of approval. And those studies themselves don't need to be very large or last very long.
Okay, well, we've reached the end of our time here, but David, I want to thank you so much for joining us today.
Yeah, it's a pleasure. Thanks, Kay.